Hi, y'all. This is Mr. Labonte. I'm going to be talking about stem cells. So, stem cells are cells that can be turned into other parts of the body, hence this wonderful cartoon of the snowmen who have found their stem cells in the snow. All right, snow women. Snow women too. Sorry about that. All right. We can talk a little bit about, there's different ways to think about stem cells, but one way you can think about them is you could look at where the stem cells are coming from. So we have embryonic stem cells, and so they would be coming from embryos, mouse embryos for mice or human embryos for humans. So these cells are what's called plurally potent within the first 7 days when the organism is called a blastula. And they can turn into any human tissue. Totipotent cells can also form the placenta, but we don't really care about making placentas, but plurally potent stem cells are the ones that can turn into any human tissue. Once you're born, you still have stem cells in your body, but these are called multipotent stem cells, and that means they can go and turn into lots of things, but they can't be turned into everything. So, for one example of what we think is probably a very strong multipotent cell is cord blood that's taken from the umbilical cord. Another one that all of us have are these hematopoietic stem cells that exist in your body in your bone marrow. You all have these, and then the discovery of these and the use of these was key in addressing all kinds of lymph cancer. So we might talk a little bit about that later. But again, so stem cells are cells that can turn into a bunch of other tissue. For your purposes, you have planaria or the flatworm. Right now, think about it. Are the flatworm cells multipotent or are they plurally potent? What's your evidence? So I'll just leave that there. All right, I'm going to talk a little bit just about the hematopoietic stem cells so you can get a specific example of how this works. So this is this stem cell, the hemocystoblast. This lives in your bone marrow, and this cell differentiates, so it divides through mitosis. So you've learned about mitosis. It divides, and as it divides, some of the DNA is closed up, and this puts it then on a particular path. Remember, every cell of your body has all the DNA necessary to make anything, but it's not all being used at the same time. So some DNA gets closed up, which turns this cell into, but first, I don't even know how to pronounce that, protheriablast, then a urethroblast, and then finally a erythrocyte. So this moves it and turns it into a red blood cell. Different types of DNA get closed down, and these turn into granulocytes. So these are white blood cells, right? So we learned a little bit about these when we were looking at SARS-CoV-2. Different DNA gets closed down. You end up with lymphocytes, and then different DNA get closed down, and this is thrombocytes are the blood cells that clot. That this is the clotting mechanism that make the clotting mechanism. So from this one type of cell, you basically have all of the different cells and some of the proteins when you look at the clotting mechanism that we need in our blood. All right, so in our blood, that's not the only place where we have stem cells. There are some stem cells that are in our brain, but frankly, that's not the primary trait of brain. As you know, I think we've talked about, and you have a sense of, we have a number of stem cells in our skin. We're continually losing our skin. Happily, we have them in our heart muscles and our skeletal muscles. So if we tear muscles, our muscles die, then they'll grow back slowly, but they will. But then also, and this is connected to the skin, right? Because remember, the outside and the inside of us are basically the same tissue. In our gut, we have stem cells. And these are these little pink cells down here. So we have these stem cells they divide, and then as they divide, basically they push. It's like a little conveyor belt. They push the cells forward until you get these cells that are on the intestinal lumen. The job of those cells, as we looked at first semester, is to allow for molecules, some type of either sugars or proteins or things like that to go through. But then eventually those cells die and then they slough off up here at the top. So this continually happens. And this is called a crypt because these cells are kind of protected, these stem cells. Their only job is just to make more cells. Okay, so these are the cells that are in our body. These are these multipotent cells that we have. But one of the reasons that we're all excited and interested in stem cells is because as we age, remember in the mitosis conversation before, our cells are going to start breaking down. They're going to start falling apart. And if we have cells that don't continually divide, then we're going to get old and wrinkly like me. So the goal would be to try to figure out, is there a way that we can make new stem cells? Not necessarily just to reverse aging, but to look at significant issues that have happened in the body and to try to address those. So disease, some type of major tissue damage, something like that, where it would be good to replace the tissue with our own tissue. So that brings us to Shinya Yamanaka, Shima Yamanaka, he's actually an MD, a surgeon, and he apparently wasn't very good at being a surgeon, and also started a lab. And he dealt with a lot of different cancers, and thought, I wonder if there's a way that we can address cancer by making stem cells, and that is kind of the holy grail of all of this. And so he looked at the difference. So we have, I'll do stem cell here, right? And then we have differentiated, DIFF, differentiated cell here, right? And then the question is, what's different about this differentiated cell and the stem cell? What are some essential differences? And there are a lot of differences. There are hundreds of differences. But some of those differences are specific proteins that are present. So there are specific proteins that are present in the stem cell that are not present in adult differentiated cells or mature cells, whether you're an adult or a kid. So what Dr. Yamanaka did is, the first thing he did is he found these 4 particular proteins and he put them in adult mice cells and let those mice cells replicate generation after generation. And those differentiated cells eventually, so these are different offspring of these cells, had cells that ended up having all of the characteristics of these pluripotent stem cells. So just by adding these proteins, he was able to take a differentiated cell and turn it back into a stem cell. Then there's a mechanism which we can talk a little bit about in class. There's different ways of doing it, but you can actually change the DNA. So you can basically unwrap the DNA, and then when you unwrap the DNA, you're going to make these proteins again, because if these are proteins, they're made by DNA, right? Then these would be called induced pluripotent stem cells. So this is the mechanism to create stem cells out of the same tissue as the organism itself. So this was a huge change, because prior to this, the way that the research that was being done with stem cells, so let's say this is person A, so this would probably be they would have some kind of an issue, or let's say it's a mouse, really. So it's mouse A. It looks like a person. We can call it whatever we want. They would have some kind of an issue. And so the way that you would get a stem cell is you would take a fetus, right? A blastula of very, very early stages. You would take the cell out of that fetus. So this is the cell. You then would take the nucleus out of that cell. And then you would insert the nucleus of the person. So this would give you all of the different types of chemicals that are in that cell, so all of the proteins and things like that that are in that cell, and that will induce pluripotency, but what it will give you is this individual. So because you have the DNA of that individual there. So what Dr. Yamanaka came up with was a way to basically to deal with the fact that to get rid of the ethical issues that some people find related to fetuses, and to have and this whole process of pulling the nucleus out of a cell and putting another nucleus in a cell. All right, so this image talks a little bit about how you get stem cells, and this is from embryos. So let me see if I can change my color here. Yeah, there. So from embryos. So from 1 to 5 days, the embryo is dividing, and this is going to make both the placenta. So that part is going to be the placenta, and this is going to be the organism. At this point, that is in fact when they are totipotent. Now these cells here, which are going to become the organism, are plurally potent. This is where you would collect these cells. You can take them now, put them on a petri dish, expose them to various types of proteins, okay? And depending, because cells listen to the cells around them, right? So if they're exposed to the proteins that are in muscle tissue or around muscle tissue, they'll turn into muscles, they'll turn into nerves, they'll turn into pancreatic islets. Important, this is one of, soon, certainly in your lifetime, probably in the next 10 years, we will probably resolve diabetes, genetic diabetes. So the potential for using stem cells to help solve significant human diseases is close. However, with induced pluripotent stem cells, I don't know if this, with induced pluripotent stem cells, there's an issue because they don't know when to stop. So this next table will show you some of the. So first we have, this is embryonic stem cells, and these are the pros and cons, okay? First of all, there is no research that's been done where we have taken embryonic stem cells and been able to put them in a human so far and regrow tissue. When you replace the nucleus, right, I'll write N replaced here, there's no rejection, okay? And that could be patient-specific, obviously, because you would put the nucleus of the person there. The ethical issues, in addition to harvesting embryos, which and I'll probably talk about that in class a little bit, but that's what I would call a straw man is the term. That is it's kind of a fake issue. But it is possible that you can do cloning because you could create what's called a germ cell. And when we get into meiosis, we'll talk about that. Fertilized cells, so fertilized egg. These, again, plurally potent. There's been a lot of research done. If you don't change the nucleus, obviously you would have immune rejection. And both with induced pluripotent stem cells and fertilized stem cells. Oncogenic potential, that means cancer, okay? So the issue here is you put these stem cells into mice, because this is not happening with people yet, um, and they don't know when to stop growing, so they create cancer. And then adult stem cells, there's been a lot of research on them, but obviously they're going to have recognition proteins that are going to make the immunological mismatch problematic. And obviously they're going to be multipotent, all right, MULTI. They're multipotent, not pluripotent here. So they're only going to be able to differentiate into some things. So this is a quick description of what's going on with stem cell research. I hope it's helpful.